The importance of meiosis is that it enables genetic diversity. Unlike the asexual form of cellular division, meiosis allows chromosome pairs with the same genes, homologous chromosomes, to exchange pieces in a process called recombination. This enables chromosome pieces from the female parent to combine with those of the male
parent. Alright. The first phase in meiosis is prophase 1. During prophase 1, the nuclear envelope breaks up and disappears. The nucleus also vanishes. The chromosomes become visible as they shorten, cool, and thicken. Also, the spindle apparatus begins to extend outward from the two centrosomes, which move to the opposite ends of the cell. Each chromosome is composed of two sister chromatids containing identical genetic information. During most of meiosis, the sister chromatids remain attached to each other at the centromere.
Metaphase 1. After prophase 1, crossing over is complete. The tetrads move to a plane called the metaphase plate, halfway between the two poles of the cell. Next, the spindle fibers attach to the centromeres of each chromosome. Both kinetochores of each sister chromatid pair are turned toward the same pole. As a result, both kinetochores attach to spindle fibers from the same pole. This is a major difference between meiosis and mitosis. It causes the two members of each chromosome pair to be separated from each other during the next stage of meiosis, anaphase 1.
During this stage, the cell starts to lengthen. The two homologs of each chromosome pair separate and move toward opposite poles drawn by the microtubules of the spindle apparatus. This contrasts with mitosis, where the sister chromatids of each homolog separate and move toward opposite poles. At this stage in meiosis, the chromatids remain together as one complete replicated chromosome.
At each pole during this stage, there is a complete haploid set of chromosomes, but each chromosome still has two sister chromatids. A cleavage furrow appears, and by the end of this stage, the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis. In some organisms, nuclear envelopes appear briefly at this point, but in others, the daughter cells begin immediately to prepare for the next meiotic division.
In prophase 2, the two daughter cells produced by the first meiotic division. As in prophase 1, the chromosomes are descended. During this stage of spermatogenesis, the cells are called secondary spermatocytes, or during oogenesis, secondary oocytes. If there was an interkinesis, then the nuclear envelopes begin to break down again during this stage. The centrosomes have replicated and are moving toward the poles.
Metaphase 2. In metaphase 2, the second stage of meiosis 2, in each of the two daughter cells produced by the first meiotic division, which are known as secondary germ cells. The spindle again draws the chromosomes to the metaphase plate. This time, unlike metaphase 1, the two kinetochores of each centromere bind to the spindle fibers from opposite poles.
Anaphase 2. During anaphase 2, the third step of meiosis, the sister chromatids of each chromosome separate and move toward opposite poles. Once they are no longer connected, the former chromatids are called unreplicated chromosomes. As the chromosomes are dragged along by the spindle apparatus, their arms can be seen dragging along behind so that the chromosomes form V shapes. The poles themselves move further apart as cytokinesis begins and the cell lengthens.
Anaphase 2. During anaphase...
No, I just read anaphase 2. You gotta go to anaphase 2.
Telophase 2. During telophase 2, the fourth step of meiosis 2, the chromosomes reach opposite poles. Cytokinesis occurs. The two cells produced by meiosis 1 divide to form four haploid daughter cells, and nuclear envelopes form. When telophase 2 is over, the two cells are entirely separated and their nuclear membranes are fully formed. Meiosis is then complete.
It's supposed to be pink and blue.
I think we got this. Get those right there. What are those?
And meiosis is complete.